Thank you very much. Can you see my screen correctly? Just checking.
Yes, all fine.
Perfect. So thank you for the introduction, and thank you to the organizers for inviting me. Since this is a CCP4 workshop, I'm going to pick up a little bit on Aaron's tone and guide you through a little bit on a teaching ground about sample delivery and protein triggering. So picking up on a couple of topics from the previous talk into a little bit more detail. So overall considerations, as Aaron said, we're always worried about sample availability because we're constantly refreshing our sample. And time-resolved experiments actually normally require a significant amount of samples. So maybe you start here where, oh, hello, there's a band of my protein, and eventually you'll get to trying to figure out how to express it, what kind of sample do you need? Do you need microcrystals? Do you need it to be in solution? And by then, maybe you start getting some crystals, you start optimizing to make microcrystals if you're going for a diffraction experiment. And in our cases, actually, we had some experiments that required quite a lot of sample. And here's an example of some microcrystals in suspension for one of our experiments. And I would like to point out that we do have a little paper coming out in ACTA-D this year regarding microcrystallization in ammonium sulfate. So if you're interested, please have a look at that. And then, of course, Aaron said, what kind of X-ray source are you going to use? And I'll get into that a little bit more later. And how are we going to synchronize our sample and our protein activity to make sure that we see what we want to see. So delivering samples, and here are some pictures to go with the last talk. So there are numerous types of sample delivering systems. Some of them are compatible with synchrotrons and X-files, for example, fixed targets, and they come in different flavors, big fixed targets, smaller fixed targets that can even go onto a goniometer. The tape drive, which is basically a type of conveyor belt system. It's very interesting, but I'm not going to have time to go into it. High viscosity extruders, which I'll point out a little bit later again, where your crystals are basically embedded in a kind of a toothpaste-like consistency stream, and this gets extruded and taken to the X-rays. And then we come more to the fluidic side, and for X-files we use liquid jets, where your sample is coming down and it's flow focused in air and comes out of a nozzle into a very thin stream. And this is necessary because, of course, X-files are extremely bright, so the sample gets exploded. And in terms of microfluidics, we can also have enclosed fluidics. And these are dedicated for synchrotrons, because if you try to put one of these on an X-file, it will just be burned straight away. So in terms of a comparison of methods, they're all, of course, different. They will consume sample differently. So in general, and this is a very, very general view of it, if you go from a fixed target to a GDVN, you will increase your sample consumption. Most of them will be capable of introducing laser activation. Mixing is a different story. So usually for fixed targets, that's a no, although of course, as you will see in a later talk today by Pedram, there's new ways of doing mixing in fixed targets, which is very exciting. High viscosity extruders, it's not really possible because of the consistency of your medium. And then we can always couple microfluidics upstream and part of designs of GDVNs and enclosed fluidics to do mixing. The total time that it takes for you to collect some data depends highly on your beamline hardware, on your sample loading. So how many crystals are we actually getting to the X-ray beam, and your sample refreshment rate. And this can be anywhere from a few minutes to hours. And again, so at some point last year, we also did a little comparison of some current serial synchrotron experiments with these different methods that you can have a look at if you'd like to get an idea of how long it might take you to collect some data. But it's always important to run some preliminary experiments because this varies wildly. Okay, so sample activation methods. As Aaron said, there's a lot of different types of motions that we can observe in proteins. They vary widely in terms of length scales and time scales. And what you want to do is to make sure that you're triggering your sample and synchronizing it quickly enough that you can see the transition that you want to see. Unsurprisingly, the easiest way to do this is to go after a protein that is naturally photoactivatable, because that means that it will actually react to light. So you can shine light on it, and these processes are usually really fast, and then just let it run and probe it at a different time. Now, only about 0.25% of proteins are naturally activatable. So of course, this is going to bring us into my most recent field of research, which is how can we actually trigger and synchronize. So there are other methods. There are some temperature jumps, which also use lasers, where you basically populate some different states of your enzyme by increasing temperature. There is mixing, which mostly is done through microfluidics. And there's another type of laser activation coupled with chemistry called Photocaging, and I will get to that a little later. The important part is that all of these different methods yield different observable structural changes. So you have to think about this when you're thinking about your system and how you're going to deliver your sample to your beam. So let's start somewhere. Let's start with mixing. So very simply and very pictorially, here is an ensemble of molecules. You add a ligand and something happens. And I think this is a fairly simple concept. It does get a little bit more complicated. So microfluidics, as I said, can be enclosed or coupled to liquid jets. And usually we use something called laminar flow. I'm not going to get into details, but this is a very controlled way of diffusing a ligand into a sample. And it is relatively low sample consumption compared to another method that I'll come to. So normally the easiest and most simple setup that we have is a T-shaped mixer where your sample comes in on one side, your ligand comes in on the other, and then what you see here is the ligand in red basically diffusing into the sample in the middle. So as you move along this line, you have an increase in your time delay. So you move your X-ray beam down your stream and you should have a different time point in your measurements. There are ways to couple this to the method. So here's an example where we have a more complicated mixer, but you see some crystals flowing down, and then we have some gas inlets. And what it's doing is creating a liquid jet that ejects out of this microfluidic. So this would be suitable for a fell experiment. And we, as I shown before, we have also produced some enclosed fluidics where it's a similar T-shaped situation here. And then you probe along this line, and the yellow part is capped on tape, which is X-ray transparent, so to reduce background. There's, of course, turbulent flow methods. So this is more like a standard stop flow system. It is fast, it is a little bit less controlled, and it tends to consume a little bit higher sample, but there are setups for this too. And then, of course, you'll hear about later about droplets onto fixed targets during Pedram's talk. So let's talk about time resolution. So this is the scheme that I had before, which is basically you have your ensemble, you add something and you trigger your reaction. And a lot of people look at this as it being almost instantaneous. And in reality, what you're doing is you're diffusing something into your ensemble of molecules. So you will start the reaction from the outside in, and you will have a time resolution issue. This can be a big issue, it can be a small issue, it really depends on your system. So mostly you're depending on your diffusion speed and the length travel on through your sample. Larger ligands will always diffuse slower, and we're talking in certain cases 1 to 2 orders of magnitude difference. And then here is the big problem, is that diffusion is also slower in viscous media. So to all of you crystallizing proteins right now, I'm sure that you have seen PEG, and maybe you have some glycerol in. All of these precipitants, they will change your diffusion rates, and it may cause you problems. Large crystals, of course, as Aaron mentioned, you can get strong diffraction, but you will have a slow, well, not a slow diffusion, but a longer length of diffusion through the crystal. And in general, if you have a 1 to 2 micron, maybe you can go to some milliseconds, maybe you're in the millisecond time scale. But as soon as you start going 1 or 2 orders of magnitude, then your time resolution really, really decreases. And this is something to keep in mind. In terms of coupling fluidics to mixing, one of our biggest enemies forever is clogging. As soon as you start increasing the size of your crystals, you can run into problems with this. So everything has to be really tested before. And signal to noise. So this is something that we were also worried about for the fluidics, which in the case, so this is a schematic of one of our devices, and it's drawn to scale. So this was the size of our crystal compared to the size of our fluidic system, and the X-ray beam is going through. And in reality, sometimes things really work out, and even under these conditions, we looked at our scattering profiles for the background, and we had very little counts, mostly around the water ring. And even a crystal this size in this kind of stream gave us really good diffraction that we could collect. So, but again, something to keep in mind. Okay, so what if mixing doesn't work? And your protein is not photoactivatable. So then we start playing some other tricks. And I'm a photochemist, so I do a lot of photochemistry dedicated to time-resolved crystallography. And that basically means that what I do is I make chemical modifications to either a protein or its ligand by adding a group that then I can cleave off with light to trigger the reaction. So it's kind of an artificial way of making something photoactivatable. And this is a little schematic. You have your inactive protein with a photocage group on, you shine some light, you take the group off, and you activate it. In terms of the chemistry, there are different groups that you can put on, and these will define your time resolution. And I will show you a little bit more, but some of them, they can be taken off on the nanosecond time scale. Some of them can be taken off on the millisecond time scale. And again, you have to think about what it is that you want to see. So photocaging considerations. These are parahydroxyphenols. They're very fast. They have short wavelengths. So they are small and they are soluble, so they have certain advantages to them. Then orthonitроbenzols are the most well-known groups, but they tend to be much slower. Although we can play around a little bit with these R groups to make them faster and also change the maximum absorption wavelength. And coumarins are definitely bigger. They're a little bit trickier to synthesize sometimes. They are fast, they have long wavelengths, long absorption wavelengths, but they tend to be a little bit less soluble. So as you can see, all of these properties, they kind of change between the photocages. So they're never one size fits all. So we can start playing around with the groups, and we usually what happens is we have a trade-off between our quantum yields, and this comes back to how much of this are you triggering, what percentage of your molecules are you triggering, and your extinction coefficients. And then of course, you have to have a friendly chemist with you, because sometimes making these things is really not easy. So sometimes you can buy them. So for example, there is photocaged ATP available for purchase. And we did use this on a time-resolved serial, a time-resolved scattering experiment, which I don't really have time to go into much details, but basically we pulsed the system, and what we were trying to see was binding of ATP and then a dimerization event. And here's a little video of our laser setup. And we could collect data. This was collected ID 09 at ESRF, and we did see over time the formation of a dimer. We could fit our radius of gyration to get kinetics, which is an important part of the time-resolved experiments. And looking at the differences in curves, we could even see some initial changes that we couldn't really model, but we thought could be related to ATP, the ATP binding event, which was very exciting. So this was the first real demonstration of photodecaging coupled to a time-resolved scattering experiments. So what happens when you can't use photocaged ATP or one of the few other things that are available commercially? Well, you basically dedicate a research group to that. So this is my lab here. It is yellow because we make photoactive compounds. So we had to get rid of a certain number of wavelengths. We have a fully equipped lab basically to make these things. And just to show you that, as I said before, sometimes this isn't easy. So one of the projects that we're working on is towards studying the dynamics of G protein coupled receptors. These are membrane proteins. They're very hard because there's a lot of medications that target these proteins. And in a collaboration with Arizona State University, we're making some photocage ligands. So the ligand is down here called BI16707. And as I said, different cages have different properties. We know that we don't need to go very fast, but we want to make sure that we have the correct cage. So we're actually making 2 different compounds at the moment to test in a house for all of the spectroscopic properties. And it's a little bit of a long synthesis, but as I said, we have a dedicated lab, so this is what we're doing. Why did we decide to do this? Well, the crystals grow in LCP, so mixing is just not possible. Sample can be limiting, so also using fluidics could be a little bit of an issue in this case. What we can do is really soak our photocage ligand in and then just trigger it in situ. So for us, this was the correct approach, even though synthetically it's a little bit challenging to get there. It's definitely worth putting the time and effort to design the experiment in this way to get it to work. So just to finish off, I would like to just point out that all of these experiments are a multidimensional problem. You have to think about your sample, you have to think about your time resolution, you have to think about your signal to noise, but there are a lot of methods. There's a lot of things available to you, a lot of beamlines, a lot of expertise. So there's always a way around it. And the real question is, what can we see? What do you want to see? And really, when you're going to run some assays to figure out your kinetics, a lot of us usually call that you should try to run those assays in the final experiment. And that means it really comes down to, again, these questions about diffusion rates and et cetera in your different buffers. So if you can, try to figure out a way to run some preliminary assays in the same conditions that you're going to use for your final experiments. So I'll leave you with some quick acknowledgements. This is our socially distant group photo because, of course, pandemic, so we had to be 6 feet apart from everyone. Emmanuel Moa is the postdoc in the group doing most of the synthetic chemistry, and Linda Kristen joined us. She's my biolab technician, and she's a rock star, really. I have several collaborators here in the building, and here's a list of a lot of other people that were involved in these other projects that I glanced over time. And just to finish off, I would like to say that we do have a couple of positions open through BioXFL and HWI, and the BioXFL conference is going to happen at the end of the month, and it's still open for registration if anyone's interested. And with that, I will finish my talk and take any questions.
Super, thanks very much. Just quickly trying to get as many in as possible.